Hello, my name is Suzanne Duce. My poster is about a school workbook I have developed that contains several web-based bioinformatic exercises suitable for 15 to 18-year-old biology school pupils. Each exercise is designed to reinforce concepts that are being taught in the classroom in topics such as molecular biology and genetics. I am a member of the Jalview team. Jalview is developed in Professor Jeff Barton's group in the School of Life Sciences at the University of Dundee, with funding from the BBSRC and the Wellcome Trust. Jalview is one of the most widely used programs for visualizing and analyzing DNA, RNA, and protein sequences. It has a multiple window interface that allows scientists to read files directly from public biological databases and view sequences, 3D structure, and evolutionary trees. Jalview also has a number of tools for aligning and analyzing sequences. The workbook can be opened from the school's webpage on the Jalview website. The instructions are arranged in two columns. The first column contains text that describe each step, and the second contains screenshots that illustrate these steps. The workbook also contains additional supporting information and questions. We have tried to make the exercises as easy to run as possible. For example, clicking a link on the Jalview School's webpage will start the exercise. This launches Jalview in an adjacent web browser, reads in the files, and opens the 3D structure viewer as appropriate. The exercises run in a web browser using a JavaScript version of the Jalview desktop application called Jalview.js. This was developed in collaboration with Bob Hansen at St. Olave's College in Minnesota. As a result, the only equipment required is a computer or tablet with a web browser, PDF viewer, and internet access. Significantly, no software has to be downloaded. In Exercise 1, sequences of DNA are viewed alongside 3D structure. The structure viewer is interactive, so the double helix can be rotated and zoomed. The windows are also linked, so colouring the bases in the sequence will also colour the bases in the structure. When a nucleotide base is selected on the sequence, its location on the structure is visible. In Exercise 2, a split frame view opens in Jalview. This views the DNA sequence of a gene alongside the protein product that it codes. Placing the mouse on an amino acid residue in the protein sequence will highlight the codons in the DNA that codes for that specific amino acid. RNA can also be viewed. In Exercise 3, a riboswitch mRNA sequence is viewed alongside its 3D structure. Proteins with a range of different biological functions are also studied, allowing pupils to investigate how protein structure affects biological function. There is also an exercise that compares myoglobin sequences from a range of animals. Differences in the sequences are examined and a tree created. The workbook and links to run the exercises are available at the school's webpage on the Jalview website. Other additional materials such as videos are also available. The final set of exercises focuses on how mutations in the genome can cause disease. First, the DNA sequence of the gene that is linked to sickle cell anemia is studied, and the exons and introns identified. The next exercise focuses on the single nucleotide mutation in the coding DNA responsible for sickle cell anemia. Its impact on the beta-globin protein is investigated, and the location of the mutation on the hemoglobin protein determined. As pupils work through the exercises, they gradually develop the skills required to run Jalview, allowing them to investigate these molecules independently. The end of the workbook contains some supplementary advanced exercises where pupils read protein sequences from public biological databases such as the Protein Data Bank. Our hope is that the workbook will act as a springboard to allow pupils to branch out and investigate other biological molecules for themselves. Please do go to the website and check out the workbook and try out the exercises for yourself.